the latest update of gene-independent therapies, for RP. In most RP cases, there are no therapeutic treatments. A set of RP-causing genes has already been identified, and is of major interest for developing gene therapies. Nevertheless, depending on the nature and the state of the disease, gene-independent strategies are needed. Various strategies to halt RP patients' vision loss process or maintain function of the retina are under research. These therapeutic interventions include neuroprotection, direct reprogramming of affected optic cells, the application of non-coding RNA, artificial photoreceptors by Optogenetics and cell replacement, neuroprotection, Retrobulbar injection of neurotrophic factors, including serum and nerve growth factors, which contain a large number of active factors that counteract the degeneration of photoreceptors. They also contain the essential protein for the self-renewal of optic stem cells. The other way of neuroprotection is an orally taken supplement called StemActiv. It contains anti-inflammatory, anti-oxidative and anti-apoptosis components, thereby optimizes the microenvironment of the retina. Direct reprogramming of affected optic cells. RPE cell transplantation therapies are under clinical investigation. They showed great promise to improve visual function in blind patients. These cells are generated from human stem cells. Similar treatments for replacing photoreceptors are extensively tested in clinical models. The application of non-coding RNA. Non-coding RNA is essential for photoreceptor maturation, function and survival. This marks the start of promising new cures, taking advantage of developments in the areas of genetic engineering and stem cell research. Artificial photoreceptors by Optogenetics and cell replacement. Chinese scientists have created artificial photoreceptors to help blind mice see. The artificial receptors, which are extremely small nanowires, were implanted into the damaged retinas of mice and caused mimic vision. However, we are not that close to having artificial vision for human beings. The ability to see involves a complex process. The photoreceptors capture light from outside the eye and transform it into electrical signals. These signals travel to the brain through the optic nerve, where they become images. During recent years, major breakthroughs have been made, such as the application of optogenetic. It has partially recovered a blind patient's visual function by targeting retinal ganglion cells.